today. Okay, this particular lesson, we're going to be talking about these learning outcomes, right? So just like I've given you in the past, if you get to the end of this and you're like, oh my gosh, that was so much information, I have no idea where to even start with this. Can you explain how a resting membrane potential, a resting potential is created? Right? Can you describe the functions of these channels, and so on and so forth? Okay, we just got finished talking about selective permeability in our cells. And so the reason why I have chosen to throw neurophysiology in here is because the only way that our neurons work is if we have appropriate facilitated diffusion, we have appropriate active transport, we have appropriate exocytosis. All of those different things have to be in place in order for the function of the neuron and therefore the function of our brain. Okay, so that's why this content is right here. And so a neuron really is just like any other cell in that it has a cell body, right, which we can also call a soma. So here we can see the nucleus, we can see the RER, the Golgi apparatus, the mitochondria, right? So all those things that we've been talking about for the past several classes, they're all in this cell. So there is really just a cell that happens to have specific functional modifications to give rise to the functionality of the specialized function of the cell. And so specifically what we have here are dendrites. Right, so all of these little sticky things out here are phospholipid bilayer surrounded extensions off of the cell body. And so these are actually equipped with different types of receptors based on what this cell is sensitive to, right? So we have neurons that are sensitive to pain in our skin. We have neurons that are receptive to different chemicals. Right? So in your taste buds, you have dendrites on your neurons that are essentially able to bind to, a bacon molecule versus a sugar molecule, right? And so all of these cells are therefore specific to sensing a particular type of stimulus, right? Again, that stimulus can be something like pressure or pain, or it can be something like a chemical. And so the dendrites are responsible for actually detecting that stimulus. Okay. If this particular neuron were to detect a stimulus, what would happen is that something called depolarization is going to occur. So actually we change the permeability of the cell. If that stimulus is strong enough and the cell becomes so permeable to something or the other, what's going to happen is an action potential. So we can see up here an action potential, an electrical stimulus is going to be generated in this funnel-shaped region of the cell. Okay, so what we can see is this funnel shape called an axon hillock. Action potentials are generated there, and they are going to flow lightning fast, right? Think the speed of electricity through these or through this extension of the cell. And so these electrical stimuli travel all the way down the axon, and ultimately that message then has to be passed on to another cell, what is called a postsynaptic cell. And so that kind of conversion of electrical stimulus into chemical stimulus is going to happen down here at these little knobs at the end of the axon. They have a bunch of different names. We can call them axon terminals. We can call them synaptic bulbs. There are tons of other words, but essentially the ends of the axons are where that message gets passed on to the next cell. Okay, so ultimately these specialized structures, right, the dendrites, the receptive ends, the axons, the transmissible ends, these give rise to the function of the neuron, right? So excitable cells responsible for sending and receiving signals, and we do that with electricity. And so here is a slightly simpler image of a neuron. All right, we're actually going to be drawing something that's even simpler yet. This is, these are the dendrites. Action potentials are generated here, and then conduction happens in a single direction all the way down the axon until we get down here to the secretory region. Okay, so There we go. These cells are either at rest or they are sending an action potential, right? So at rest, these cells are receptive, right? Those dendrites are able to detect a stimulus. When that stimulus is received, then and only then does the electricity within the cell change. And so at rest, the cell actually has a particular charge to it. And so I'm going to start our little drawing here. Okay, to the best of my ability, of course. So let's first draw in some dendrites, right? I'm going to make them really big, right? We'll focus on this particular dendrite, but of course there are lots of them, okay? The cells that we've been looking at, so the neurons that we have been examining so far, actually, I'm going to draw this a little bit bigger. The cells that we have been examining so far have been the cells that you might find in the central nervous system. We're going to look at simpler cells than that, just because, for the ease of discussing this. In these cells, we have, a big old dendrite, right? Just one. Okay. And here the axon hillock. And, oops, do you see that? No. Sorry about that, guys. Again, a learning curve. And so the axon hillock is going to narrow down into a single axon, going all the way to the end of the cell. Okay. And so way down here, we can call this an axon terminal. Okay, so the end, the synaptic bulb. And here you can see that I haven't drawn in the cell body. And the reason for that is that in these more peripheral sensory neurons, the cell body kind of sticks off the axon. Okay, so this here would be the nucleus and the Golgi and all of those different things that we're used to seeing in cell. All right, so here's the receptive end. Here is the axon, which ultimately is sending this electrical signal, and all the way at the end, we have this axon terminal where we are going to see the message being converted into a chemical signal and passed on to the next cell. Now, there is a gap in between the axon terminal of one cell and the dendrite of another cell. That is called the synapse. Okay, and we're talking like nanometers wide, right? So these are actually really, really close together. Okay. As I said, we are talking about neurophysiology right now because we need to understand facilitated diffusion and active transport in order to understand the neuron. And so let's start adding some things in here. In the axon terminal, there are actually vesicles, right, which are made down here in the cell body in the soma, and they are transported via our friends, those little molecular motors, all the way down to the axon terminal. Now, those little vesicles there happen to be filled with something called neurotransmitters. Okay, so within here, we have neurotransmitters. Okay, specifically, the one that we'll talk about the most is called acetylcholine, or AC, capital C, capital A, H, acetylcholine. Okay, so again, those are housed within these vesicles. All right, the vesicles we can call synaptic vesicles. All right, I know there's a lot of terms, but we will be using these terms as we go along. Okay, so those are our synaptic vesicles. Okay, so ultimately, what we are going to see, right, when we're through this process, is exocytosis of these chemicals, of these neurotransmitters, out of the synaptic vesicles and into the synapse. Okay, so that's where we're going here. All right, we also have a series of channel proteins. Okay, so along this entire axon, we have channels that are embedded within the membrane. Right? Remember that channel proteins are proteins, therefore they bind specifically and tightly to their substrate, or in this case, they are going to allow only a particular thing, in this case an ion, to flow with its concentration gradient. Okay? These ones in green that I have drawn, we will call voltage. Actually, make the colors match here for you guys. They are called voltage gated sodium channels. Okay, and so this gate concept, right? So this isn't something that we have talked about at this point, but some of these channel proteins actually are either open or closed, and they can change given, in this case, voltage. And I, of course, spelled voltage wrong, but at rest, these voltage gated sodium channels actually have like a little ball and chain on the outside. Okay, so they are closed, right? So essentially, sodium is not able to flow with its concentration gradient when the cell is at rest. Okay, the only way for this little ball and chain to kind of fling open out to the side, okay, is when the correct voltage is going, or when the correct voltage is present within the cell. Okay. Okay. Also, we have voltage-gated potassium channels. And again, they are going to be located, embedded within the cell, all the way down the entire axon. At this point, I am only drawing these channels on one side of the axon, just so that we have a little bit more space to talk about different processes. Okay. These voltage-gated potassium channels also have a little ball and chain. Okay, they are closed at rest. Right? So when the cell is just sitting there waiting for some kind of stimulus, these channels are actually closed. Okay, so these, to abbreviate, are voltage-gated potassium channels. Okay. If you guys have any questions, just throw it out there. I'll keep the chat up for you guys. All right. There is a third and final type of voltage-gated channel, and I promise we are going somewhere with all of this. The final voltage-gated channel is only located, within the synaptic, or within the axon terminal. All right, so here we go. This is also closed, right, as you are seeing here. And this is a voltage-gated calcium channel. Okay, so as you will see as we go through the semester, calcium causes stuff to bind to other stuff. And so in this case, what calcium's role is in this process is that when calcium is allowed to flood into the cell, it is going to cause these synaptic vesicles to fuse with this little presynaptic membrane, and therefore exocytosis can proceed. And so again, that's where we're going with this. So all of these channels so far are examples of facilitated diffusion. Okay, so these are closed at rest, but when they open, they are going to allow both potassium, sodium, or calcium to flow with its concentration gradient. Now there are some channels, and so passive transport, that are always open, right? You actually did learn about these in the videos I gave you. These are called potassium leak channels. Oops, potassium leak channels. Okay, so these are always open. They're always allowing potassium to flow with its concentration gradient. Okay. So what is the gradient? You guys actually already know a little bit about this particular gradient because of those videos I gave you. All right, so also embedded within the membrane are sodium-potassium pumps. Okay, so here is a sodium-potassium pump. And I promise all of this stuff is going to come together, all right, in a kind of elegant way. Okay, so sodium-potassium pumps are always working to pump sodium out of the membrane. Right? Remember that 3 sodiums are pumped with every single ATP. And 2 potassiums are pumped into the cell with every single ATP. Right? So here we have this pumping action all the time. Okay, so pumping potassium and pumping sodium out. Okay, now as we pump this potassium in, we see that there are potassium leak channels. And in fact, there are sodium leak channels as well, but they are definitely not as abundant within this, within these cell membranes. Okay, so as the potassium is pumped in, a little bit of it is actually going to be leaking out. Okay, so we'll get into that here in just a second. Final type of proteins that are embedded within this membrane. I understand this is all sorts of things going on here. So once again, final proteins. These are receptor proteins. And there's actually a lot of them, but I'm just going to draw one because it is very challenging to draw with a mouse, sorry. All right, so these are receptor proteins. So they're what actually make the dendrites receptive to some type of stimulus. Also, these are important because they are associated with a channel protein. Okay, so what we can see here is I have drawn kind of like a channel. This is allowing some ion, a particular type of ion, to flow with its concentration gradient. Ultimately, at rest, these channels are generally closed, but as soon as a ligand binds to this receptor, the channel is going to open and that ion is going to flow into or out of the cell, depending on its concentration gradient. Okay, does that make sense so far? Or if it doesn't make sense, speak up. Okay, so these are all the players that make this process work. All right, so let's go back to our PowerPoint here. Okay, so all of those things are within the cell's membrane at rest. So before we can even, detect a stimulus, before it can even generate an action potential, the cell needs to maintain something called resting membrane potential. And so what we can see over here is the phospholipid bilayer surrounding the entire neuron. This here is a voltmeter, and actually in your lab simulation today, you are going to be using a voltmeter to detect the charge on the inside of the cell. Now, in order to maintain this charge, we need to have 2 different types of concentration gradients, ultimately leading to a relatively positive environment on the outside of the cell, so here is extracellular fluid, and a relatively negative charge on the inside of the cell. Okay? Ultimately, the membrane potential, or the charge inside relative to outside the cell, must be negative 70 millivolts. Right? So in a neuron, negative 70 is what we need inside the cell. So inside this neuron needs to be very negatively charged in order to be receptive to a stimulus. Okay? And again, this electrical difference, this electrical gradient, is created by creating concentration gradients of sodium and potassium. And as we can see here, sodium is relatively high outside, so 150 versus 15, so high sodium outside the cell, and potassium is relatively high inside the cell, so high potassium inside relative to much lower potassium on the outside. Okay, we also have some other things going on as well. We can see this A. This A is referring to anything else that's negatively charged in the cell. So you can think of proteins as being, on average, negatively charged. Also, chloride ions generally kind of hang out at this membrane here. So all of the negatively charged stuff that's in the cell is this right here. And so we can see there's a lot of negative in here and very little of these negatively charged proteins outside the cell. Okay. So how exactly do we establish this resting membrane potential to begin with? It requires sodium-potassium pumps, as you might imagine, as well as leak channels. Right? So again, there are more potassium leak channels than there are sodium leak channels, and so we only drew those into our diagram. Okay, so what's happening all the time, all around the membrane, even though I only drew it on one side of the cell, is that sodium-potassium pumps are using ATP, right, so a very energy-intensive process. They're using ATP to pump 3 sodiums out, and it's an antiporter, so at the same time it pumps 3 sodiums out, it's going to pump 2 potassiums into the cell. Okay, so here's outside, here's inside. Ultimately, we end up with a ton of sodium outside and a ton of potassium inside. Okay, so now we have 2 independent concentration gradients. Hooray. But wait, I thought we were trying to make a negative inside the cell, right? Potassiums are positive, and so too are sodiums, right? So we are pumping in positive stuff inside the cell, but somehow we have to end up with a negative cell. And so what leads to this very negative environment is the permeability of that membrane, right? The allowing of potassium ions, or the allowing of stuff to go across the cell, specifically potassium ions. All right, so just as we pump potassium in, those potassium ions are continually leaking out. Now, of course, this pump is pumping faster in than potassium is leaking out, so there's still a higher concentration gradient of potassium inside, but we get this steady trickle of positive ions leaving the cell. Okay, and so where we once had tons of positive, now we have less positive, leaving behind all of these other negatively charged things. Right? So proteins are negative, chloride ions again are going to hang out right underneath this membrane here, and so ultimately the working together of the sodium and potassium pumps and the potassium leak channels is going to lead to 2 different things: concentration gradients of sodium and potassium, and a relatively negative environment inside the cell. And so when we use this little voltmeter, right, so measuring outside, and here we poke the cell membrane and essentially see that inside is -70 relative to outside. Okay, and so we can call this neuron polarized. Right? Remember, polar means opposite, and so we have an opposite charge inside the cell compared to outside the cell. Right, so the neuron is polarized when the voltage difference across the membrane. Right, so this is a difference across the membrane. Okay, so let's go back to our little diagram here. What we can see, and I'm going to draw, or rather type in trying to type in, interesting. That is different and inconvenient, but that's okay. So kind of step 0, sorry guys, is maintaining this resting membrane potential. Oh my gosh, I'm so sorry. It's really not letting me type right now for whatever reason. But this resting membrane potential, as we just talked about, is a result of lots of sodium being pumped out, lots of potassium being pumped in, leaving a positive environment inside and outside the cell. But some of those potassiums are then able to leak out these leak channels, which are always open, right? Even at rest, they're always open. Therefore, leaving all sorts of negative stuff inside the cell and all sorts of positive stuff outside the cell. And so even though I've only drawn it down here, these processes are happening everywhere throughout the entire cell. Right? So lots of negatives everywhere relative to positives on the outside. Okay, remember, resting membrane potential is negative 70 millivolts. Okay, and so if we were to actually stick a voltmeter into this membrane at rest, we would see negative 70. Okay. Going back to our slideshow. Okay. So, again, before any of this transmission of action potentials can possibly start, we need to begin with a resting membrane potential throughout the entire cell. When a receptor binds to its ligand, so binds to you know, a hot pepper molecule or something, what that's going to do is open up channels and lead to what is called a graded potential, or a local change in the charge of the cell. If that charge is strong enough, it's going to lead to an action potential, zipping all the way down to the end of the cell, and finally leading to exocytosis of neurotransmitters to the postsynaptic cell. Right, lots of words. But let's see, hopefully, if this plays down here. What it's going to show you is sodium ions, potassium ions, different types of channels, and a pump. Nope, I guess not. So I'll walk you through this. Here is our stimulus coming in. What that does is it depolarizes, right, or increases the charge closer to 0 within the cell body, right? And then finally, we have the action potential zipping all the way down the cell. Okay, if we zoom into that neuron's membrane, we can see voltage-gated channels, right? Sodium and potassium channels. Keep in mind they are closed at rest. Okay. Also, we have sodium-potassium pumps. So here, when the voltage changes, sodium channels are going to open up, those voltage-gated channels, and therefore allow sodium to flow with its concentration gradient. At the same time, a voltage change is going to open up the voltage-gated potassium channels, which then allows potassium to flow with its gradient. Right? This action here is going to end up, or it's going to produce a positive charge inside the cell. So here we can see the negatives have changed to positives because now we have so much sodium inside the cell. This change in charge is going to lead to the next voltage-gated sodium channels in line to open up, right? And this process is going to continue. It's a chain reaction. Change in charge because of a stimulus opens up a voltage-gated channel, depolarizes the cell, makes it positive, and then this creates a current of positive ions. Now, in order to get the cell back down to resting membrane potential, we need to keep working with those sodium-potassium pumps, right, to regenerate the concentration gradients. And also, we're going to see that the role of the voltage-gated potassium channels is to actually regenerate that resting membrane potential. Okay, and so what we can see here in this final clip of the video is a voltmeter. And I'm sorry it's so blurry here, but essentially -70 is resting membrane potential. When sodium floods into the cell, this is going to lead to a spike in the charge. And so this says +30. And so that is the action potential, right? At +30, voltage-gated potassium channels open up, and this is going to repolarize the cell. That is making it negative. Okay, so sorry about the lack of volume there. Okay, so let us now look at the stimulation of the cell. Okay, so what we can see over here is a dendrite, right? It happens to be wrapped in these layers and layers of tissue, kind of like we see in our cells with those lamellated corpuscles, or in our skin with those lamellated corpuscles. Essentially, if we stimulate this dendrite, these are the different electrical signatures that we're actually going to see. Okay, so here we can see 1 voltmeter, which is this right here, and here's a 2nd voltmeter in the axon over here. Okay, and so we, in this case, are just stimulating the cell with electricity, right? So as we give the cell a little bit of a stimulus, what's going to happen is a similar depolarization of just the dendrite, right? Not the rest of the cell just yet, but just the dendrite is going to match that stimulus strength. And actually, this is very relevant to your lab exercise that you're going to be doing later. Okay, if these stimuli are strong enough, right? So if there is enough depolarization, enough positive coming into the cell, what's going to happen is that in the axon, you are going to see a series of action potentials, or at least one of these action potentials. So these local changes in charge, right? So ultimately, what the heck is happening here in the dendrite is going to change greatly depending on how long the stimulus is, right? So we can see that as long as we are stimulating the dendrite, there is going to be depolarization in that dendrite. The number of ion channels open, right? So we're going to see that here shortly. And of course, the types of ion channels that open. For the most part, what we talk about in this class is depolarization, right? So essentially here is at rest. When we stimulate the cell, we let in positive ions, and therefore the cell gets closer and closer to 0. So it gets more and more and more positive as opposed to -70. This is called depolarization because it is bringing the cell again closer to 0. There is another option, however, depending on the stimulus, you might actually hyperpolarize the cell, right? So here at rest, if an inhibitory stimulus is applied to the dendrite, what is going to happen instead is that the cell gets even farther away from 0, and therefore it is hyperpolarized. Okay, so let's see if we can watch this. If not, you guys have these videos embedded.
Before any stimulation, the neuron is at rest. Recall that the resting membrane potential of a typical neuron is -70 millivolts. This potential depends on the distribution of sodium and potassium ions across the plasma membrane and their movement via leak channels and the sodium-potassium ion pumps. Stimuli are detected by the receptive region of the neuron, which generally consists of the dendrites and cell body. When a neuron is stimulated, its resting membrane potential changes because ions begin to cross the plasma membrane through gated ion channels. If ligand-gated sodium ion channels open, sodium ions follow their electrochemical gradient and enter the cytosol. As the inside of the neuron gains positive charges, it becomes less negative, a change called depolarization. If ligand-gated potassium ion channels open instead, potassium ions follow their electrochemical gradient out of the cell, and the cell loses positive charges. This causes the membrane potential to become more negative than it is at rest, a change called hyperpolarization. Hyperpolarization can also be caused by the movement of negatively charged ions, such as chloride, into the cell. Small local changes in the membrane potential are called local potentials or graded potentials. They can be depolarizing and therefore cause positive charges to enter the cytosol and make the membrane potential less negative. Alternatively, they can be hyperpolarizing and make the membrane potential more negative. Local potentials also vary in strength, depending on the length of stimulation and the number of ion channels that open. We can compare local potentials to the ripples caused by a pebble thrown into a pond. A larger stimulus, or pebble, will cause a larger ripple effect, and a smaller stimulus, or pebble, will cause a smaller ripple effect. Another important feature of local potentials is that they are decremental, meaning that they weaken as they get farther away from the source of stimulation, and so are only capable of traveling over short distances. If we use our pebble analogy again, as ripples move away from the point where the pebble hit the water, they diminish in size and eventually disappear. For this reason, local potentials cannot be used for long-distance signaling. A single neuron can receive many incoming stimuli in the form of depolarizing and hyperpolarizing local potentials. These local potentials combine as they spread across the membrane of the neuron. The sum of these potentials determines whether or not an action potential is generated. We will discuss that effect in the action potential animation that follows.
Okay, glad that worked, hooray. So as the video clip just said, these graded or local potentials are only hanging out in that particular spot, right, where the stimulus actually was. And so we have local potentials occurring when a ligand binds to a receptor protein. Oh, it's just not going to work. Bummer. Okay, so step one in generating an action potential is for a ligand to bind to a receptor. Right, so here is a bacon molecule, right, binding to the receptor. When that happens, this channel protein is going to open up. And therefore, if this is a stimulatory, or an excitatory stimulus, which it is in this case, this channel is going to allow sodium to flow into the membrane. So sodium is, okay. Remember that sodium is super high outside the cell. And so sodium is going to flow down its concentration gradient into the cell. Just like the video clip just showed, this influx of sodium is going to add positive charges to the local area of the cell. Remember that these things are kind of rippling out, right? It is decremental, so essentially the charge is going to kind of phase out over time. But if you have enough bacon molecules, right? You just ate a big piece of bacon, right? And so this binding of the ligand, binding of the bacon to the receptor is going to happen all over the dendrite. So you're going to have these receptor potentials, right, adding together, right? Again, if there is enough of a stimulus. Okay. So this first step here is just the stimulus binding to the receptor. And that's all I'm going to fight through here. Okay, so let's go back to our presentation. Okay, so what next, right? At this point, we just have a couple graded potentials. They are only hanging out in the local area. We don't actually have an action potential yet. And so the way that we get an action potential, right? So this lightning fast message sent all the way down the axon, right? And keep in mind that sometimes these cells can go from the tips of your fingers all the way up through your spinal cord and into your brain, right? So several feet long of a single cell. And so we have to maintain this electrical signal that entire length. a graded potential is not going to do the trick. And so what we can see in this graph here, right, this is essentially the reading from one of those voltmeters. This is time on the x-axis. On the y-axis, we have the membrane potential. And so we start at rest, right? This particular cell was receptive to the bacon molecules, right? One molecule of bacon bound, or was able to bind to the receptor, that depolarized the local area of the cell just a little bit, but not too much, right? Another bacon molecule binds in a little bit, not quite enough, right? And again, but if you get multiple bacon molecules binding to multiple receptors at the same time, you get a huge influx of sodium, right? So that is going to depolarize the cell quite substantially. And so what we can see is that when depolarization occurs to a certain point, right, it's going to surpass, or at least achieve, this threshold value, right? This much depolarization, not enough. But once you get this much depolarization, then and only then are you going to initiate this chain reaction of actually opening up these voltage-gated sodium channels, right? So think about the fact that these are called voltage-gated. So in other words, you need a particular voltage, right, or a threshold voltage within the cell in order for these things to open up. Okay, and so ultimately this leads to a positive feedback loop, right? Once one voltage gated channel opens up, that causes more depolarization. That's going to open up the next channel and the next channel and the next channel. And so at last we have achieved generating an action potential. Okay, so let me just, we'll go back to our diagram. Okay. So step one, we stimulated the cell by binding a ligand to a receptor on the dendrite. This led to an influx of sodium, causing a graded potential, a local increase in charge in the dendrite only. Now, if enough of this depolarization kind of adds together, a threshold value is going to be achieved, right? And so that threshold is actually the voltage at which these voltage-gated sodium channels, particularly here within the axon hillock, are going to fling open their gates and therefore allow for the influx of sodium into the cell. Okay, so step number 2 here. Once threshold is achieved, right, and usually threshold is, depending on the cell, it's like -55, -60, -40, depending on the cell again. But once a threshold voltage is achieved, what is going to happen is these, oh, we're just drawing today, okay. These voltage gates are going to fling open. And remember that we have tons of sodium all around on the outside of the cell. And so now sodium floods into the cell, taking this formerly negative environment and now making a positive environment, right? Remember that positives and negatives are attracted to each other. And so these positives are essentially going to flow as a current down towards these other negatives throughout the rest of the cell. Bringing with them this depolarization, right? Now, this particular area of the cell is now positive, is now above the threshold at which these voltage gates are going to fling open, okay? All right, so again, once these voltage gates fling open because there is a positive environment inside the cell, more sodium floods into the cell, making the next area of the cell positive, right? So this is going to continue, essentially creating a lightning fast transmission of this electrical or this positively charged stimulus all the way down the axon, ultimately leading to a negative charge within the axon terminal. Okay, so Step one is generating graded potentials. They are not good for super fast, super long distance transmission of a signal. But step number two, threshold is achieved. We have generated an action potential, right? An action potential, AP for short, which is this chain reaction of depolarization all the way down the cell lightning fast. Okay. That makes sense, hopefully. It's the real deal, right? All right, so here is our action potential. Keep in mind that when you're looking at these graphs here, so these readings from voltmeters, these short little blips here are graded potentials. Action potentials shoot up really fast, so really steep slope. Generally, they top out about positive 30. Okay, and so here is just another way of showing this. Graded potential, if the graded potential achieves threshold, what's going to happen is voltage-gated sodium channels open up, leading to a rapid influx of sodium, and therefore the local area of the cell would then be positive 30 millivolts. Right? We can see down here, again, these are different moments in time. So this is the beginning. A few milliseconds later after that, we generate this positive environment by the influx of sodium. Positive ions, so the sodium ions slide to the side, right? Therefore making the next area of the cell positively charged, right? Same thing, sliding over until ultimately this positive charge has slipped all the way down the cell. Okay, again, super fast. This is an action potential, not a graded potential. What I also want to point out here, and that's the next step that we're going to talk about, is the fact that the cell returns back to its original environment after the action potential has passed. Okay, so here it goes back to being -70, or actually for a little while, a little bit lower than -7. Okay, so we at this point have only talked about the voltage-gated sodium channels, but the voltage-gated potassium channels are equally important. All right, so at the top of this action potential here, right, we have just flung open those gates of the sodium channels, and all of a sudden we see this very quick about face in the charge, right? So it gets super positive and then goes right back to being super negative again. So what's happening here is that this approximately positive 30 voltage is going to cause two things to happen. First of all, it is going to inactivate those sodium ion channels, right? So, all right. Essentially, that little ball and chain is going to come back and block up the pathway for sodium to enter the cell anymore, right? So now we're not having an influx of positive, right? That in and of itself is going to let the cell get a little bit more negative. But also at the same time, positive 30 millivolts is going to stimulate these potassium channels, again, voltage gated. They are triggered by a particular voltage, in this case, positive 30. They're triggered to open up. Remember that potassium is super high inside the cell, and so if we open up these gates, what's going to happen is that potassium is going to flow out with its concentration gradient, so there's an efflux or a mass exit of potassium outside the cell. So now we have stopped letting positives come in, and we have let more positives come out, therefore returning the cell to its more negative state. Now, if you take a look all the way down here, you can see that yes, the cell has returned to its negative state because of the closing sodium channels, opening potassium channels. What we can also see is that there's a little bit of an overshoot, right? So here's -70, that's where the cell needs to be at rest, but the cell actually gets a little bit more negative for a little while before returning to its true resting membrane potential. And so what's happening here is that these potassium channels are a little bit sluggish. Okay, so When they get to about threshold, they're going to start closing their gates, but it takes a little bit longer for those gates to close. And so they're actually letting more potassium leak out of the cell than they absolutely need. And so this leads to a hyperpolarization. Right? So actually farther from threshold than it normally would be at rest. Okay, let's watch this. I'll watch from the beginning.
As you learned previously, a single neuron can receive many incoming stimuli in the form of depolarizing or hyperpolarizing local potentials. In a process called neural integration, all of these incoming stimuli are combined. If enough of the stimuli are depolarizing, their cumulative effect will reach the trigger zone. This zone consists of the axon hillock and the initial segment of the axon. When the trigger zone depolarizes to threshold, it generates an action potential. An action potential is a large, uniform depolarization followed by a rapid repolarization. Action potentials are all or none events. In other words, if an action potential is triggered, it will occur and will propagate down the axon. If, however, the trigger zone does not depolarize to threshold, no action potential will be generated. You can think of an action potential as a bullet being fired from a gun. If you pull the trigger with enough strength, the gun will fire the bullet. Once the trigger is pulled, the shot cannot be undone. In a similar way, an action potential, once fired, will rapidly travel down the axon without losing any strength and cannot be reversed or stopped. Let's now examine the events of an action potential more closely. We begin with the axolemma in the trigger zone depolarizing due to the summation of local potentials. It depolarizes from the resting potential of -70 millivolts to a threshold potential of -55 millivolts. At the threshold potential of -55 millivolts, voltage-gated sodium ion channels open. Sodium ions quickly rush into the axoplasm, driven by their electrochemical gradient, and cause a rapid depolarization to about +30 millivolts. At the peak of depolarization, the voltage-gated sodium ion channels inactivate, and sodium ion influx stops. If we look closely, the sodium ion channels have two gates, one called the activation gate and one called the inactivation gate. The gate that closes and plugs the channel at +30 millivolts is the inactivation gate. At the same time, voltage-gated potassium ion channels open and potassium ions exit the axoplasm, moving with their electrochemical gradient. Now the cell has stopped gaining positive charges and is losing positive charges instead.
Okay, let's add this step into our diagram. Okay, so now we are, we're at step number 3, right? Our action potential has shot all the way down the cell. This is the rapid depolarization. Now, at the same time, right, once in any of these areas of the cell, once the positive environment has passed, what's going to happen is that the potassium ion channels are going to fling open their gates and again allow potassium to flow with its concentration gradient outside of the cell, right? Therefore allowing potassium or positive ions to leave, and so the cell in this particular area would then return back to its original negative area. Okay, so this is the repolarization of the cell. Okay, and again, that is happening in a similar chain reaction all the way down the cell. Okay. Yeah. Okay, so the action potential really only takes a couple milliseconds, right? So very fast. something that you wouldn't actually consciously be able to experience. But we can zoom into the action potential and actually break it down into a couple different periods. Specifically, when those sodium and or potassium channels are open, this is called the absolute refractory period. Right? So essentially, as soon as the cell reaches threshold, sodium channels are open. Right? Which means that if you try to trigger another action potential in that cell, right, at this particular moment in time, you can't physically open any more sodium channels, right? Sodium channels are open or they're closed, right? So you absolutely cannot generate another action potential as long as the sodium channels are already open. Similarly, you cannot generate an action potential, even if you try to trigger one here over and over and over again, really, really strong stimulus, you cannot generate another action potential because the potassium channels are still open, right? You can't depolarize a cell if potassium is leaking profusely from that cell, right? So really strongly pulling the cell back down to negative, right? So as long as the cell is above threshold, absolutely no more action potentials can be generated. Right? Action potentials are all or none, just like the video said. They're either there or they're not. You can't generate another one at the same exact time. You can't generate a stronger action potential, right? So you would never see an action potential way up here at positive 50. They tap out at positive 30 no matter what. Okay. Now, once the cell is repolarized to below threshold, in theory, you can generate an action potential at this time, right? So if you try to trigger a cell right here in time, you can do it, but it is relatively difficult to do so. That is, the sodium channels are, once again, able to open up. So hooray, you can do that, but there still is a little bit of potassium, a little bit more than normal potassium, leaking out through those voltage-gated potassium channels. Remember, they're a little bit sluggish to close back up. And so you can think about kind of how difficult it would be to bring the cell up to threshold from down here, right? So from this hyperpolarized area, it would take a lot stronger of a graded potential to bring the cell up to negative 55 if you're starting at negative 80. Right? So this period right after the action potential, again, like a millisecond, really not that long. But this is called the relative refractory period because it is relatively difficult to generate another action potential. Okay. Once again, action potentials are all or nothing. They are able to travel very quickly down an axon, travel a long distance, and in fact, we'll talk about another evolutionary modification to even increase this ability even farther yet. They're self-propagating, right? Once the bullet is fired from the gun, it is going to go, right? In the same way, once threshold is reached, the action potential is going to go. Okay? Self-propagating. Okay, and so then what? All right, we're not quite done with this story just yet. At this point, we have sent an electrical stimulus down an axon, so from the tip of your finger all the way up through your spinal cord and into your brain. But how exactly does this message get relayed to the next neuron, to the postsynaptic neuron within your brain? And so we've all seen animations of a synapse before. So they're often on medication commercials online, right? Particularly ones about depression, or treating depression. And so this is a presynaptic neuron, this is a postsynaptic neuron, and all of this stuff happening in between is the release of neurotransmitters. And so the only way we get the release of neurotransmitters, the natural way anyway, is to complete exocytosis. So bulk transport, also an active process, so it takes lots and lots of energy, but this release of chemicals into the synapse is going to ultimately allow these molecules to diffuse across this very, very tiny space. bind to receptors on the postsynaptic cell's dendrite membrane. And I'll say that again, the postsynaptic cell, so this one right here, the one that's receiving the message, dendrite, the receptive end of the cell, and of course the membrane here, the sarcolemma, or the exolemma, sorry. Right, so the way that we get this synaptic transmission, this exocytosis of neurotransmitters into the synapse, is that same exact depolarization. So the action potential is going to arrive finally at the axon terminal, and that depolarization is going to trigger the opening of our final voltage-gated channel that we'll talk about today, the calcium channels. So voltage-gated calcium channels are going to trigger to open up, allowing calcium to flow with its concentration gradient into the synaptic terminal or the synaptic bulb. As I said earlier today, calcium generally causes stuff to bind to other stuff. And in this case, calcium is going to cause these synaptic vesicle membranes to fuse with the presynaptic membrane. Right? So it was its own independent vesicle. Now calcium is going to trigger this membrane to bind to this membrane and therefore open up, dumping its contents out into the synapse. Okay, so we're going to add that into our diagram here. Once again, this positive charge has finally ended up way down here in the axon terminal. We are going to see the calcium channels open up. Calcium floods into the cell. And as I said, calcium causes this synaptic vesicle membrane to fuse to the presynaptic membrane, right? And as a result, these neurotransmitters, in this case, the acetylcholine, is going to flood out of the neuron into the extracellular space of the synapse. And so that is step number 4, right? So exocytosis, one of our bulk transport mechanisms. Exocytosis. Once again, I'm really sorry, I don't know why I can't do anything but draw right now. So exocytosis of neurotransmitters, these guys, out into the synapse. Ultimately, these molecules are then going to diffuse, right, from the area of high concentration to low concentration, right? So you spray the perfume, you can ultimately allow those molecules to diffuse to their area of low concentration on the other side of the room and smell them way far away from where they already were. And so here's the high concentration. These molecules diffuse across the across this space, and ultimately they are going to bind I really cannot do it. Bind to a receptor on the postsynaptic cell. Right, so the neurotransmitters are now the ligands. The ligand, of course, binds to the receptor, opening up its ligand-gated sodium channels, right? So opening up these associated channels, allowing sodium to flood into that cell, causing a graded potential, these nice ripples of positive charge. If enough acetylcholine binds to these receptors on the postsynaptic cell, this is going to lead to yet another action potential in this postsynaptic cell. Okay, so once again, some kind of a stimulus triggered this receptor, caused an influx of sodium. This caused a graded potential, a depolarization. It was strong enough to achieve threshold, and so these voltage-gated sodium channels opened up, allowing an influx of sodium into the axon. This created a current of positive charge, so positive 30, leading to a chain reaction of opening voltage-gated sodium channels all the way down the length of the axon. Simultaneously, once that action potential reached positive 30, the sodium channels closed and potassium channels opened, leading to an efflux or an exiting of potassium and ultimately a return to resting membrane potential, a repolarization. Okay, when the action potential reaches the axon terminal, this leads to an influx of calcium, right? So opening of these voltage-gated calcium channels. This influx leads to the binding of the synaptic vesicle membrane to the presynaptic membrane and an exocytosis of acetylcholine. Acetylcholine then diffuses across the synapse, binds to the postsynaptic cell receptors, leading to a graded potential and an action potential if that signal is strong enough. Okay, so when you guys are making your videos, your open-ended response videos, which again are going to count towards your exam 1 grade, you can do something very similar to what I just did. Don't forget to talk about resting membrane potential, right? So in order for any of this stuff to work, we need to create 2 gradients and we need to allow a little bit of potassium leakage. Okay, so to be thorough here, number 5 is this is going to stimulate the next cell. So the postsynaptic cell, this can be another neuron, this can be a gland, it could be a muscle, all sorts of different things. Now, essentially, as long as these neurotransmitters are located within the synapse, they're going to keep on binding to these receptors and therefore keep on triggering action potential after action potential. And so if we don't clean up the synapse, right, if we don't get these messaging molecules out of there, essentially this stimulus could be long gone, but we're still telling the next cell that, you know, we just had bacon, we just had bacon, we just had bacon. And while that sounds fantastic, right, to always have the, you know, delightful feeling of just having something tasty, it's not healthy, right? And so ultimately this postsynaptic cell will become desensitized, and therefore you will never be able to experience the joy of eating bacon again because you're pretty much going to take these receptors off the membrane. And so it is so very important to clean up the synapses all the time. And in fact, a lot of those medications that are treating depression or schizophrenia are really just affecting how long these neurotransmitters are going to stay within the synapse. So that's beyond the point. We'll talk about that more later this term. But our cells have a recycling mechanism. Okay, so essentially there are these little enzymes that are located within the synapse that are actually going to break down the neurotransmitters, right? And for the most part, the pieces of neurotransmitters actually get taken back up into the cell via a process called endocytosis. So actually engulfing these extra little pieces and taking them back up into the cell so we can reconstruct them into neurotransmitters. Okay, so this here we'll look at on a slide here in just a second, but this is the action of something called acetylcholinesterase, and there's no way that I am going to try to spell that out with a mouse. Okay, so acetylcholinesterase is going to recycle those neurotransmitters so that the stimulus is temporally linked to the message, right? So at the same time, hooray, and also so that you don't overstimulate those cells. So what this video is showing you before we get into acetylcholinesterase is a synaptic vesicle that is filled with acetylcholine. Okay, it is being taken by our friends, the molecular motors, all the way down the cytoskeleton fibers, the microtubules, all the way to the synaptic bulb. And so ultimately, we want to release all of these things out into the synapse at the appropriate time. And so the only way that that happens is when calcium, right, all these little blue guys around here, is going to actually allow the vesicles, as we'll see very soon here, to essentially dock with the presynaptic membrane, right? So here calcium is allowing the docking, membranes fuse, neurotransmitters ultimately released out into the synapse. Now, all those details are not something that you need to be able to produce for me, particularly within that open-ended video response that you guys are all responsible for doing. but I thought it might be helpful for you guys to actually see how calcium causes stuff to bind to other stuff. Right. To be continued, it will come up again. Okay, so when we were looking at our little diagram that we were drawing, we left off talking about the synaptic bulb, allowing calcium in, the docking of these synaptic vesicles, and ultimately the release of acetylcholine. Acetylcholine leads to a depolarization on the postsynaptic membrane. So that's kind of where we're looking here. As soon as acetylcholine binds to these receptors, acetylcholinesterase, okay? So ACE, it's an enzyme. Acetylcholine, right? And it's essentially the ester, like, you know, breaking these apart into esters. Ultimately, acetylcholinesterase is going to break this neurotransmitter into multiple pieces, right? And part of those pieces, the choline part specifically, are going to be taken back up into the cell. So endocytosis here, put back together by a product of the mitochondria, actually a byproduct of cell respiration, and ultimately put back into a vesicle so it could be released once again. And again, that's really important. We really don't want extra neurotransmitters hanging out in the synaptic cleft within the synapse. Essentially, a lot of drugs like cocaine and nicotine ultimately promote the longevity of neurotransmitters within the synapses. Just, you know, essentially how they work, but this is why you become addicted to them, and you ultimately need more and more and more to experience the same feeling of elation or high or buzz or whatever, because your body actually starts taking these receptors out, and so you need to throw more and more at them. Okay, again, that's besides the point. All right, as you've seen in a couple of the video clips I've shown you so far, each neuron that we have is actually receiving all sorts of different messages from other cells. So here we can see synaptic bulbs from tons of other axons, tons of other neurons. Each one of these can be triggering the cell to get closer to the threshold or get farther from threshold, right? So we've seen that in the video clip so far, but I do want to add a couple terms here. Big clunky terms, but they should make sense. Excitatory postsynaptic potential, or EPSP. These excite the cells. They get the cell closer to threshold, right? Potential is charge over a membrane, right? So is it negative? Is it getting more positive or more negative? And postsynaptic, right? So this is essentially the receptive cell. So these would be the postsynaptic membranes and the postsynaptic cell. Okay. On the other hand, inhibitory postsynaptic potentials are going to hyperpolarize the cell. Right? So it is really important for you guys to start getting fluent with using terms like polarization, depolarization, hyperpolarization, repolarization, right? Postsynaptic, presynaptic, excitatory, inhibitory, all of these different terms are going to become important. A, because you're making a video about this process, but B, you're going to have to read questions about them on your lecture exam, which is scheduled for Monday. And to actually, like, right away know what these words mean is really going to help you to perform well. Okay, before we move on to what these funny looking things are around the axons, I do want to point out that yes, these postsynaptic cells are receiving information from tons of different neurons all at the same time. And to actually achieve threshold, it usually involves multiple excitatory stimuli, either occurring at the same time or occurring in relatively the same place on that cell. Ultimately, your neurons are summarizing all of the information that they're getting from all of these different cells. Right, so this one down here is receiving both inhibitory and excitatory cells. They sum up all of this information, and essentially, if the excitatory stimuli are strong enough to get to threshold, there will be an action potential. Okay, one final time looking at this, this graph here, it is really important. I can guarantee that there are going to be questions about it on your lecture exam. Hint, hint, nudge, nudge. you should be able to know what's happening at each of these different steps, right? So this is a graded potential. This is when the sodium, voltage-gated sodium channels are opening up, causing depolarization. This is when those sodium channels close, potassium channels open, leading to repolarization, getting more and more negative. Both of these steps here are within the absolute refractory period. You cannot stimulate another action potential as hard as you try. Relative refractory period are when those potassium channels are sluggishly closing, and so you can technically generate an action potential. It's just really hard because of this hyperpolarization. Okay. So again, that graph, super important, and it's broken down for you here. Again, hint, hint. Okay, so at this point, I've been talking about just how fast these action potentials are. And really, all of this is happening lightning fast. We do not realize when action potentials are firing just because they are so fast. They are beyond our ability to perceive. However, they're not all created equal. There are ways of varying action potential conduction speed, right? So how quickly the action potential shoots down the axon to the axon terminal. Just like wires, just like your garden hose versus a fire hose, the diameter of the axon, or the diameter of the hose, the diameter of the wire, is going to determine the rate of flow of electricity or water or whatever. In general, wider substances, wider hoses, the fire hose is going to have less resistance, right? There's less surface area to kind of create friction of the water, or for the water. And so there's less resistance with these wider pathways than there would be with these thinner or smaller diameter. And so what that means is that with less resistance, flow is going to be greater, right? So in terms of, in terms of water flowing, right, the fire hose is going to have lots and lots more flow than your tiny little faucet in your kitchen, right? Just because of that difference in diameter. Same thing with electricity. Skinny wires have more resistance, and therefore the electricity simply isn't going to travel as quickly. And so neurons are essentially wires. They're essentially hoses. Wider axons are going to produce less resistance, and therefore flow of these ions is going to be much quicker. And so for what we would consider to be more simplistic organisms like a squid, and a squid isn't a random choice on my part. We have learned so much about how our neurons work by studying squid. Squids don't really have a lot going on up there, and so they have pretty much 2 giant axons to control everything within their entire body. And so they don't need to have skinny axons, right? They have huge axons. Yay, we can study them. Yay, these action potentials can travel from the, it's called brain, but it's really not, within the squid, all the way down to the other end of the organism, right? So hooray. The problem is when we get more complex, right? So obviously we as humans would like to think of ourselves as being much more complex. We have a lot of different regions of our brain, neurons, in order to process information, remember information, right? Produce emotional states, not just like glandular secretion and movement, right? We have tons of stuff going on within our body, and therefore we have a lot, or we have a need for a lot of neurons. And so this is actually an image of the brain, highly simplified, but these are all axons, right? So you can't physically fit massive diameter axons, right? So we as humans, and of course lots and lots of other animals, more complex animals with more complex functions and organs, we had to fit more neurons in the same space, and so we made them much skinnier. Now, as I said, skinnier pathways, right, smaller diameter wires, hoses, whatever, have more resistance and therefore less flow. And so our skinny little axons for the most part, should have really slow flow of current. Right? And so our evolutionary response to this problem, right, our advantage is something called myelination. Right? So essentially, we can increase the conduction speed of our action potentials by insulating those wires, those axons. And so this substance called myelin is actually a fatty substance located within specialized cells. When these neuroglial cells, so when the helper cells actually wrap around the axons, it is insulating in such a way that it increases the transmission rate of those action potentials. Okay, and so what we can see here is the cell body, the soma, the dendrites, and here is the one single axon. Wrapping around those axons is actually a cell, right, called a Schwann cell. We'll get into that in a moment. But this cell is chock full of that fatty substance, which is a great insulator, called myelin. And so the cell is just going to wrap around and around and around and around the axon. You can think of a roll of paper towels or a roll of toilet paper, right? The paper goes around the cardboard over and over and over again. In the same way, these neuroglia, these Schwann cells, are going to wrap around the axon. Okay? The structure that is created by this cell is called a myelin sheath. And in fact, this makes the axon appear to be white. And so later, for exam number 2, so starting on Tuesday next week, we're going to talk about white matter versus gray matter. Essentially, the white matter is all of these insulated axons, right, with lots and lots of myelin sheaths. Okay, and so we can see here, myelinated axons conduct action potentials significantly faster, right? So 15 to 20 times faster than unmyelinated axons. And that's how we can get so many neurons packed within our brains without being super slow about it. Okay. Just a second here. I would rather the computer talk to you about this video here. So we'll get to that here in a minute. Okay. This video, short video, is going to show you what type of transmission we have been talking about, and then it's going to show you what is much more common within our cells.
When an action potential in an axon spreads to a neighboring region of its membrane by a series of small steps, the process is called continuous propagation. When it propagates by jumping from one site to another along the axon, the process is called saltatory propagation. Saltatory propagation occurs along axons that have myelin sheaths. In the peripheral nervous system, these myelin sheaths are formed by Schwann cells. The myelin acts as an electrical insulator, allowing ions to move across the cell membrane only at the gap or node between adjacent Schwann cells. Therefore, action potentials rapidly travel from node to node.
Okay. So we have been talking about continuous conduction of action potentials. Saltatory propagation essentially adds these myelin sheaths, which insulate the axons in such a way that the action potential can almost literally jump across this insulated region of the neuron and essentially increase that speed. And so before we go back to the presentation, I want to show you this fun little demonstration in slow motion. Essentially, these dominoes are going to represent more or less those voltage-gated sodium channels opening up, right? So, of course, once one opens up, it opens up another one, another one, another one. And so this here represents continuous conduction, right? In order to get the action potential all the way down here to the synaptic bulb, you need to literally open up every single one of those sodium channels. Right? It seems like it wouldn't really take that much time, and in fact it doesn't, it's just milliseconds, but we can increase the speed by these piece of paper right here, these represent the myelin sheaths. And so what you're going to see is the action potential is going to literally jump over, right? The energy is going to be, and movement is going to be transferred by this entire sheath here to the next region of the neuron, transferred, and finally to the end. So again, continuous conduction versus saltatory conduction. Okay, so hopefully now you can appreciate just how much more quickly myelin sheaths make synaptic or make action potential propagation. Okay, so come back to that. There are two different types of cells that are responsible for myelinating axons in the peripheral nervous system. So pretty much anything but your brain and your spinal cord, we use Schwann cells. Note that one Schwann cell is one myelin sheath. That's it, nothing else fancy. In our central nervous system, we have a lot more densely packed neurons, and so we actually use one cell, all right, so one cell called an oligodendrocyte to myelinate multiple parts of the same, or multiple parts of the same axon and multiple axons. Okay, so again, Schwann cells in the periphery, so these would be in your leg, these would be in your brain and your spinal cord. And so this here is showing you what is happening inside that axon. Right, again, with continuous, that's exactly what we've been talking about so far. And that's in fact what I want you to describe in your open-ended video that's due in a few days. Right, so this channel opens, depolarizes the cell, opens this channel, depolarizes the cell, and so on and so forth. Every single channel has to be opened up. Saltatory conduction, on the other hand, insulates the axons in such a way that essentially this electricity, this current of ions actually can just shoot right through this insulated region and then open up channels here, right, in this space between the two myelin sheaths, and so on and so forth. So literally the action potential is leaping across these myelin sheaths. Okay. And so another way of looking at this is in this way right here. What this little white line is showing you is the size of the voltage. So essentially, how strong is that depolarization, right? So the reason why we can't just depolarize a local area of the cell and have that voltage change, like go all the way to the end of the cell, is because these localized graded potentials get smaller and smaller and smaller the farther from the initial stimulus they are. And so what you're doing when you actually are conducting an action or propagating an action potential with continuous conduction is that you are continually regenerating that action potential, right? So here the initial stimulus, really strong depolarization, but because those ripples get fainter and fainter and fainter the farther away from the stimulus you go, you have to regenerate that action potential. So then you open up the next voltage gated channel, increase the strength of that stimulus. Stimulus gets a little bit weaker, and then you don't want to fall below threshold, of course, and so then you would open up the next channel and so on and so forth. Okay, and so literally continuous conduction involves continuously regenerating that action potential because the strength of the stimulus, the strength of the depolarization gets smaller and smaller over space. Okay, and so when we add myelin, right, how is that changing this process? Well, essentially, it's not something I've talked about so far, but there is a bit of leakage of these ions, right? So the reason why, or another reason why it's getting so small so quickly here is that some of these ions are actually able to leak out the membrane, right? And so when we add these multiple layers of myelin, it is really difficult for these ions to leak across the membrane. In fact, it is easier for these ions to flow through the axon than it is to flow out of the axon. Okay, and so essentially this gives us a little bit more time, a little bit more space, a little more distance, which over which this action potential can travel before we have to regenerate that action potential. And so we have these channels only located within the spaces between the myelin sheaths. These spaces are called nodes of Ranvier. So a French word, so Ranvier, not Ranvier. I've heard all sorts of different pronunciations, but node of Ranvier. And so here is where we are regenerating the action potential, not within this insulated region. Right? So just like the first little video clip showed you, it literally looks like the action potential is leaping because there's less leakage through this, this cell right here. Okay. We can appreciate, as always, this is kind of a theme of AP science in general, we can appreciate just how important a structure is because we know what happens when it goes wrong. So essentially, that's how it's supposed to work, right? Action potential leaping from node of Ranvier to node of Ranvier. There is a condition, an autoimmune disorder, in which your body attacks the myelin sheaths, specifically the myelin sheaths within the central nervous system, so your brain and your spinal cord. What this does, or what this results in, is a lot more leakage of those ions through the axon membrane than there really should be. And so by the time the action potential gets through what should be myelinated, the strength of the stimulus has degraded so very much that what might ultimately happen is that the cell falls below threshold, and therefore it won't open up these next voltage-gated sodium channels, and essentially the action potential is stopped. And so this is a big deal, right? If you are sending an action potential, you have some kind of a message that you need to get to another area of the body. This can be, recalling memories. It can be, forming decisions. This can be commanding your muscles to work, right? So, grab the pen, right? And then you can't grab the pen because the message never actually gets from your brain all the way down to those muscles. And so, right, the symptoms here are progressive slowing of the action potentials, which can result in sensations, right? So different stimuli, messages never actually get into your brain. Changes in behavior and cognitive abilities, right? Again, these oligodendrocytes are located within your brain. And so if your brain is not able to send messages or not able to get messages to their final destination, all of a sudden you can't do the things that you can normally do, including move. Okay? So again, we know the importance of a structure when that structure is no longer functional. And so in the lab, we can kind of remove these structures to see what's important, or unfortunately, we can take a look at diseases and see